Hello students, welcome to the CET classes of biology. So we are all meeting after a very long time, and I hope you're all doing good with your health. And you have completed your final examination, second PU board examination, and I congratulate all of you for doing well in the examination. And today we are going to see the first mock paper of biology, KCET biology. So before I start, I would like to clear here. So of course, you know, the paper contains 60 questions, and maybe we will be covering 30 questions in one session. And the other 30 questions will be considered for the next session. Each question will carry 1 mark, right? And see, you should be very clear that here the questions that we are going to consider for this KCET will be of little easier. So if you compare the questions with the NEET examination, the NEET questions will be of the next level. So these will be of a little lower level. And most of these questions may be directly asked from the NCERT, or maybe they are taken from the NCERT, but they have been twisted in such a way that they may not look like NCERT questions. But so it is necessary for you to be thorough with the NCERT syllabus in order to answer CET questions. Right? So let us go to the first question here. See, I'm not going to talk much about the synopsis here. So we are going to solve the questions directly, and so as and when required regarding the particular question, we are going to talk about in detail there, right? So let us look at the first question. Which chromosome contains the least number of genes? Right? So you have all studied that chromosome 1 has the most number of genes, accounting for 2968 genes present on the chromosome number 1, whereas chromosome Y will consist of least number of chromosomes, lowest number of chromosomes, that is 231. So which is the answer? Obviously, answer is chromosome Y. Right? So I hope you're all seeing these statements. This is the part where I'm going to give the explanation for the question, and so answer will be marked with this blue arrow, right? Yes. So second question we can go for. So a typical angiosperm embryo sac at maturity consists of a typical angiosperm embryo sac. Embryo sac is also called as what? Embryo sac can also be called as, it is also called as what? See here, polygonum type of embryo sac anta kuda kariteve adakke alva, because a typical angiosperm embryo sac, which is also called as polygonum type of embryo sac, will be mainly consisting of how many? 7 cells and 8 nuclei. Correct, no? So, or the female gametophyte anta kuda karibodu adakke. And how did these 7 cells come, and why there are 8 nuclei is because, see, we also say that the female gametophyte in the angiosperms is monosporic in development anta kariteve. Monosporic means what? See, one of the cells of the nucellus. You can imagine the structure of the ovule, right? Within the ovule, at the center of the ovule, there is a mass of undifferentiated cells. That is what is called as nucellus, right? In that nucellus, just one cell, Yella cell alla, onde ondu cellu, which is slightly near to the micropylar pole, which is slightly near to the micropylar opening. That cell will grow bigger in size, and that will be called as what? Megaspore mother cell anta kariteve. That megaspore mother cell will undergo meiotic cell division. Right? That means one cell undergoes meiotic cell division anta helidre, it's going to form 4 megaspores. And out of those megaspores, 3 are going to degenerate and only 1 will be retained. That one haploid megaspore that is retained after the degeneration of 3 other megaspores, that one retaining megaspore will later develop into what? The female gametophyte or the embryo sac. So within that one haploid megaspore, a series of free nuclear divisions are going to take place. So how many free nuclear divisions are going to take place? 4 free nuclear divisions are going to take place, forming 8 nuclei. 8 nucleus formation agutte. After the formation of 8 nuclei, wall formation takes place in such a way that they form 7 cells. Andre ಈ 8 ನ್ಯೂಕ್ಲಿಯಸ್ಗಳಲ್ಲಿ so there must be one cell which is consisting of 2 nuclei. Which is that cell? That is the cell that is present at the center. You can look at this diagram. So central cell ಅಂತ ಕರೀತೇವೆ. Central cell ನಲ್ಲಿ 2 ನ್ಯೂಕ್ಲಿಯಸ್ಗಳಿರುತ್ತೆ and these 2 nuclei are called as polar nuclei because so one of the nucleus comes from this pole, the other nucleus comes from this pole. That is why they are called as polar nuclei, and this cell that is consisting of 2 polar nuclei will be called as central cell. Right? So this complete set of cells is called as egg apparatus anta karitave, which will be consisting of the egg and 2 synergid cells. Right? So you know the properties of synergid cell, functions of synergid cell. So I will not go into the details of it because that you have learnt in the double fertilization event. And these 3 cells at the chalazal end will be called as antipodal cells. So what will be the answer here? Will be 7 cells and 8 nucleated condition. Right? So this is monosporic in development. This is also very important for you to remember. The development of the female gametophyte or the embryo sac in a typical angiosperm. Right? So that is what is monosporic in development. We can go for the next one. Identify the wrong statement among these. Alleles. So IA and IB. So hege odbodu ನೀವು. So A and B produce a blood group of AB. Correct, no? So you have studied about the blood grouping from this chart. So ಇಲ್ಲಿ ನೀವು ನೋಡ್ತಾ ಇದ್ದೀರಿ. So allele from parent 1, allele from parent 2. So when both of them are consisting of A genes on the A allele on the I genes, so definitely it will form the A blood group. A ಮತ್ತು B ಎರಡೂ ಒಟ್ಟಿಗೆ ಇದ್ರೆ, so it is because of the codominance the blood group will be AB blood group. Right? And one is dominant and the other is recessive. The one which is dominant will get expressed. That is why here it is A. Similarly, it is in this case it will be. So it is again codominant. B and A will be again AB. And B and B both are dominant. Definitely blood group B. And only B is dominant and the other gene is recessive. And that is why the blood group will be B. And so if both the genes are recessive, then blood group will be O. So this one chart you have to understand. This chart should be in your mind and you can answer so any number of questions like of this type, like based on the blood grouping. So the first statement says that alleles A and B produce a blood group AB. Right? So this is correct according to the law of codominance. Both IA and IB are present together and they express because of codominance. So this can be the reason for the first statement. Both statements are correct. Third statement, alleles B and C also produce the blood group AB. Is it possible? That means they are taking the recessive genes here, recessive B and recessive C. So what it is going to become? When they are recessive, obviously it should be O blood group. Right? So this is the wrong statement. When look at the fourth statement, when IB and B, that means one is dominant, other is recessive, or so you can also consider I is present, only IB is expressed. This is correct. The one which is dominant will be expressed. So answer is what? Third one is the answer. Going for the next question, fourth one, the codon AUG has dual function. It is the initiation codon. So this is one function they have already given here and also code for. That means you have to say or you have to identify out of which of these amino acids will be coded by this particular codon called AUG. So you should know the chart here. ಈ ಚಾರ್ಟ್ ಅನ್ನು ಸ್ವಲ್ಪ ನೆನಪಿಟ್ಕೊಳ್ಳೋಕೆ ಪ್ರಯತ್ನ ಮಾಡಿ. So ಇಲ್ಲಿ AUG can be an easy one. ನಿಮಗೆ ಗೊತ್ತಿದೆ AUG has dual function. It is also called as a start codon and it will code for methionine ಅಂತ ನಿಮಗೆ ಗೊತ್ತಿದೆ. I hope you know, right? But similarly, ಇದರಲ್ಲಿ ಬೇರೆ ಯಾವುದು like CUU codes for ಅಂತ ಕೇಳಬಹುದು. It is coding for leucine, or maybe ACU codes for which one? Thrombin. So like this. So you may have to remember the chart. So try to remember the chart, and the answer for this is what? Methionine. Right? So this is the only codon having the dual functions. Right? So at least ನೀವು ಕಂಪ್ಲೀಟ್ ಚಾರ್ಟ್ ಅನ್ನು ನೆನಪಿಟ್ಕೊಳ್ಳೋಕ್ಕಾಗದೇ ಇದ್ರೆ ಸೊ ಅ ಫ್ಯೂ ಬೇಸಿಕ್ ಥಿಂಗ್ಸ್ ಅಬೌಟ್ ದ ಚಾರ್ಟ್ ಅಂತ ಹೇಳಿದ್ರೆ, ಒಂದು AUG ಸ್ಟಾರ್ಟ್ ಕೋಡಾನ್ ಡ್ಯೂಯಲ್ ಫಂಕ್ಷನ್ ಬಗ್ಗೆ ನೆನಪಿಟ್ಕೊಳ್ಳಿ. ಇನ್ನೊಂದು ಸ್ಟಾಪ್ ಕೋಡಾನ್ಸ್ ಯಾವುದು? ಸ್ಟಾಪ್ ಕೋಡಾನ್ಸ್ ಯಾವುದಿದೆ? ಸೊ UAA, UAG and so here you can one more you can consider UAA, UAG and UGA, right? So here So these three codons you need to remember: UAA, UGA, and UAG. At least ಅಷ್ಟನ್ನಾದರೂ ನೆನಪಿಟ್ಕೊಳ್ಳಿ ಪ್ರಯತ್ನ ಮಾಡಿ. Going for the next one, rheumatoid arthritis is. So this question is coming from the chapter Human Health and Disease. So you need to identify what kind of disorder or disease is this. Is it an allergy or autoimmune disease or genetic disorder or infection? What kind of disease is this ಅಂತ ನೀವು ಐಡೆಂಟಿಫೈ ಮಾಡಬೇಕಾಗಿದೆ. So let us go for the explanation here. Sometimes due to genetic and other unknown reasons, the body attacks its own cells, right? So body is going to attack the phagocytes, or the immune system of the body is going to attack on its own cells, and that is what is called as autoimmune disease. And if it becomes a disease, if it becomes a disorder, genetic disorder, then it will be called as autoimmune disease anta kariteve. And rheumatoid arthritis is one such autoimmune disease. So answer is autoimmune disease. Going for the sixth one, pleurobrachia. Pleurobrachia belongs to the phylum. See, this is coming from the animal kingdom chapter. You will have to remember the examples of different organisms belonging to different phyla, right? You will also need to remember a basic classification of many different examples. Maybe belongs to which class anta kelbodu. Yavado ಒಂದು organism ಅನ್ನು name ಮಾಡಿ it belongs to which class anta kelbodu, which phylum anta kelbodu, right? So it is better you know the basic biological classification of at least the examples which are mentioned in the textbook in CET. So now coming back to this question, pleurobrachia belongs to the phylum. See, you have seen this diagram of pleurobrachia. So it is an example of ctenophora, right? So they also are known for, ctenophores are known for what? Bioluminescence anta ನೀವು ಕಲ್ತಿದ್ದೀರಿ. And you should also know the characteristics of, I'm not going into the details of the characteristics of the phyla here. So ctenophora is the answer. Now, seventh one, with respect to eutrophication. So eutrophication anta ಹೇಳಿದ ಕೂಡಲೇ what comes to your mind is the natural aging of a lake, right, or a pond, any fresh water or body anta ಹೇಳಿ. So with respect to that, the statements X and Y, we have to analyze these statements. It is a natural process of aging of a lake. Yes or no? It's a natural process of aging of a lake. Not to get confused, if in case in the place of eutrophication, if they had mentioned cultural eutrophication anta ittu anta helidre, then it will be what? It is not a natural process. It will be like artificial, or there will be the participation of human beings. Yes or no? So that is why we are talking about eutrophication here. It is a natural aging of lake. So statement X will be considered as true statement. Statement Y, it is finally it should be leading to the revival of the lake. That means once again the lake will be renewed. So because of this statement says that what? Because of eutrophication, the lake revival takes place. Is that true? No. Here, because of the process of eutrophication, actually the death of the lake is taking place here. So that is why, so this statement Y is not true. And which can be the answer? Answer is 3. Only statement X is true and Y is false, right? So going to the next one, 8th one. The seed of which of the following is known for the longest dormancy period? Dormancy means you already know it's a resting phase or a sleeping state of the seed, just to overcome the unfavorable conditions. Especially seed ಗೆ ಬೇಕಾಗಿರುವಂತಹ favorable condition ಅಂತ ಹೇಳಿದ್ರೆ, it should get the proper temperature and moisture. ಇದೆರಡು ಇದ್ರೆ then seed can readily germinate. But until the seed gets that favorable condition, it has to be in a certain state of resting, and that state of resting will be called as dormancy, during which in some seeds there will be the development of a very hard seed coat. ಅದೆಲ್ಲ ನೀವು ಕಲ್ತಿದ್ದೀರಿ. Now in this, which one of these plants is known for having the longest dormancy period ಅಂತ ನಾವು ಕೇಳ್ತಾ ಇದ್ದೀವಿ? Phoenix dactylifera, ಖರ್ಜೂರ, right? Lupinus arcticus. So lupinus, it is belonging to the family Solanaceae. Mangifera indica ಅಂದ್ರೆ mango ಅಂತ ನಿಮಗೆ ಗೊತ್ತಿದೆ. So both 1 and 2 are the given options here. Now actually the answer is both 1 and 2. That means both Phoenix dactylifera and Lupinus arcticus. So here Phoenix dactylifera was found in the King Herod's palace, and that was found to be 10,000 years old, and Lupinus arcticus also was found to be 2,000 years old. So during an excavation, these were found, and they are known to have the longest dormancy period here. Going to the next one, the funnel-shaped part of the oviduct closer to the ovary is called. The funnel-shaped part of the oviduct closer to the ovary is called. We are talking about the female human reproductive system, female system of the humans, reproductive system of humans. So you can look at the diagram here. So here, these are the ovaries situated, and so we are talking about the what oviduct. Oviduct alli ಮೂರು ಪಾರ್ಟ್ಗಳು ಇರುತ್ತೆ. That is the what infundibulum, ampulla and isthmus anta heli, and we are talking about what the funnel shaped structure. Which is the funnel shaped structure? That is infundibulum, which is very close to the ovary. Yes, so ampulla, isthmus, the questions, many questions can be framed on this particular oviduct. At which place exactly the fertilization takes place anta ಒಂದು question barbodu. So ampulla, isthmus junction anta kaltidiri. So be aware of these questions. I hope you are very well aware of this diagram. Going to the next one, which of the following is not a point for successful beekeeping? So beekeeping ige yavdu successful point alla. Let us go on reading the statements. Catching and hiving of swarms. So catching and hiving of swarms anta helidre, so bees anna hitkondu bandu ಒಂದು ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಹಾಕಿ, that means we are going to have a bee hive there. So man-made beehive irutte, so we are going to rear there. So this is proper one. Intermittent disturbance of the beehives, handling and collection of beeswax, knowing about the nature and habits of honeybees. See, idralli intermittent disturbance anta helidre, once you put the bees into that hive or box anta en karitivi, beekeeping box anta en karitivi. So if you put the bees into that once, you should not be disturbing that until a certain period of time. So if you keep on disturbing it again and again, the bees will get disturbed. The process of formation of honey will also get disturbed. That is why, so this one can be the wrong statement. The second statement is the wrong statement regarding the successful beekeeping practice. 11th question. A nucleoside is, see, this is about the nucleic acids. So nucleic acids, there are two different types of nucleic acids, DNA and RNA, deoxyribonucleic acid and ribonucleic acid anta kariteve. And in both of these cases, the base, or you can say the backbone of the nucleic acid, will be formed by what? The pentose sugar. Pentose sugar, there are two types of pentose sugar here. If you are talking about the DNA, it will be deoxyribose sugar. If you are talking about the RNA, it will be ribose sugar, right? And nucleoside anta helidre, on this pentose sugar there will be the attachment of, you can see, the nitrogen base, right? The nitrogen base is attached to the pentose sugar with a bond called N-glycosidic bond. To which carbon atom it is joined? To the first carbon atom. Yes, so this is what is called as nucleoside here. And so going a little further, what is a nucleotide? Not to get confused, nucleoside is different, nucleotide is different. Idakke nucleoside ge nitrogen base mattu sugar iruvanತಹ structure ge, to the third carbon atom or the fifth carbon atom, if a phosphate group joins with the help of which bond? Phosphoester bond na ಸಹಾಯದಿಂದ. So then it will be called as what? Nucleotide. You can look at the differences between these two pictures. This is nucleotide, this is nucleoside. So which is the answer here? Answer will be second one. So that is a purine or pyrimidine base plus sugar. So this is the nucleoside. 12th one, the diagram represents, so this diagram is representing the Miller's experiment. Choose the correct combination of labeling. So these questions can be very easy. You have already practiced all the diagrams for your board examination, and you should be knowing the proper labelings of this, and I hope you can answer. So take a little bit of time and try to answer this question. Which is the right answer? Yes, the answer is the first one. So A, electrodes, B, the gases which are present in this discharge tube or this thing, NH3 plus H2 plus H2O plus CH4, then cold water will be there, boiling water will be there, C will be that, and vacuum and U-trap. This is called as U-trap, E. Next one, which among the following is not produced by the distillation of fermented broth? See, in the microbes chapter, microbes in human welfare chapter, you have come across the different alcoholic products which are produced by distillation and which are not produced by distillation. So out of these, whiskey, wine, beer and brandy, ಈ ನಾಲ್ಕರಲ್ಲಿ which one is not distilled ಅಂತ ಕೇಳ್ತಿದ್ದಾರೆ, and out of this only wine is the undistilled product, whereas other products will be distilled. Distillation actually increases the alcoholic content in the drink, and that is why distillation is required for the drinks with higher alcoholic content, whereas wine is comparatively having a lower alcoholic content, and this is undistilled one. Going for the 14th one, photosynthetically active radiation, PAR, has the following range of wavelength. What is photosynthetically active radiation? See, the Earth's surface is receiving the sunlight, and if you consider that sunlight that is received by the surface of the Earth or atmosphere of the Earth to be 100%, less than 50% of this sunlight will be actually utilized for the process of photosynthesis by all the photosynthetically active organisms, not necessarily plants. It can be bacteria also or cyanobacteria also, right? Less than 50% only, plants will absorb only from 2 to 10% of this radiations from the sun, and that radiation which is actually utilized for the process of the photosynthesis will be called as photosynthetically active radiation, and this will be in the visible spectrum of light. And what is the visible spectrum of light? Will be 400 to 700 nanometer. This is an explanation about the PAR, and the answer is 400 to 700 nanometer. 15th one, identify A, B, C and D in the given pie diagram showing the global diversity of different taxa of organisms and choose the correct option. Idralli neevu A, B, C, D yavudu anta identify madbeku. So we are talking about the biodiversity of different organisms here. So out of these 4 options, so which one holds good for this diagram anta heli? So you can look at this diagram here, at least you can answer from this now. So large part of this one, more than 70% of the invertebrates will be nothing but the insects only. Then comes the B is mollusca, C is crustaceans, and D is other small animal groups, right? So they all comprise D here. So the answer is what? So answer is the third one. Yes. Going for the 16th one, Beckane disease was reported due to. Beckane disease is also known as foolish seedling disease of rice anta kariteve, and it was actually, see, this disease anna discover madidrinda, because the disease was discovered by a scientist called E. Kurosawa, a Japanese scientist. Disease and discover madidrinda, it was also possible for him to find out a particular plant hormone called as gibberellins, right? So that is what Gibberella fujikori ಅನ್ನುವಂತ ಒಂದು ಫಂಗಸ್ ನಿಂದ E. Kurosawa discover ಮಾಡಿದ್ದು, and it was the Beckane disease, and actually Gibberella fujikori was producing the gibberellic acids, and gibberellic acids, they are not only the disease causing or harmful chemicals produced by this one, but these are also naturally produced chemicals in the plant body, right? So this is also a fungal toxin. This is also more appropriately growth hormone released by the fungal infection, right? So you need to choose the most appropriate answer here. So I hope you have followed this one. Going for the 17th one, deficiency symptoms of an element first tend to appear in young leaves. So ಇಲ್ಲಿ ಕರೆಕ್ಟ್ ಓದ್ಕೊಳ್ಳಿ ಒಂದ್ಸಲ. ಡೆಫಿಷಿಯನ್ಸಿ ಸಿಂಪ್ಟಮ್ಸ್ ಆಫ್ ಅನ್ ಎಲಿಮೆಂಟ್ ಫಸ್ಟ್ ಟೆಂಡ್ ಟು ಅಪಿಯರ್ ಇನ್ ದ ಯಂಗ್ ಲೀವ್ಸ್, ನಾಟ್ ಇನ್ ದ ಓಲ್ಡರ್ ಪಾರ್ಟ್ಸ್ ಆಫ್ ದ ಪ್ಲಾಂಟ್. It is indicated that the element that is relatively immobile, right? So which one of the following elemental deficiency would show such symptoms? You have understood from the transport in plants chapter, there are certain elements in the plant body which can be easily remobilized, right? If an older part of the plant body is consisting of certain minerals, if the leaf is about to fall off from the plant body, the minerals, certain minerals, not all of them, certain minerals stored in the plant, in that part of the plant body, will be remobilized to the younger parts. That means this older part will be deficient with those particular mineral nutrients. But there are certain minerals which cannot be transported from the older parts because they form the structural component. They may be forming the cell wall, they may be forming the plasma membrane. So structural component agirutte. That is why, so which of those minerals? See, elements most readily mobilized are phosphorus, sulfur, nitrogen, potassium. These can be easily mobilized in the plant body. Some elements that are structural components like calcium and even magnesium, they cannot be remobilized. The answer is what? So illen kelidare deficiency symptoms of an element first tend to appear in the young leaves ಅಂತ ಕೇಳಿದ್ದಾರೆ. That means young leaf ಗೆ ಹೋಗ್ಲಿಲ್ಲ, it remains in the older leaves. That means easily remobilize ಆಗ್ತಾ ಇಲ್ಲ. The answer is what? Magnesium. Going for the next one, ribosomes were discovered by. So it's a very easy question directly from the textbook. It was discovered by George Palade. And let us look at this data. Camilo Golgi in the year 1898 discovered Golgi complex. D. Dewey gave the structure and function of the lysosome. Porter discovered the endoplasmic reticulum in the year 1945. George Palade in the 1952. He discovered the presence of cristae in mitochondria, and in the year 1953, the same person, discovered ribosomes. So answer is fourth one. 19th one, identify the labelings in the given diagram of the female reproductive system of cockroach. Cockroach na female reproductive system nalli you have to identify the labelings here, right? A, B, C, D ಕೊಟ್ಟಿದ್ದಾರೆ, ಯಾವುದು ಅಂತ ಐಡೆಂಟಿಫೈ ಮಾಡ್ಲಿಕ್ಕೆ ಪ್ರಯತ್ನ ಮಾಡಿ. And look at this explanation. See, answer is actually the second one. Second one answer ಇದೆ, ಅಂದ್ರೆ A is ovary, B is uterus, C is collateral gland, and D is spermatheca. You might have not heard about the C, collateral gland and spermatheca. See, spermatheca means there will be a pair of spermatheca in the cockroach female reproductive system, also in many insects, but in cockroach it will be present in the 6th abdominal fragment, and the function of spermatheca is to receive the sperms and store them, right? And collateral glands produce secretions that cover the two parallel sides of this reproductive system, female reproductive system, where the rows of eggs are laid during the oviposition. Oviposition agbekadre egg na place madlikke jaga kodutte. So answer is second one. 20th one, in Krebs cycle, GTP, that is guanosine triphosphate, is formed in substrate level phosphorylation, oxidative phosphorylation, non-cyclic phosphorylation, or cyclic phosphorylation. Non-cyclic mattu cyclic phosphorylation are a part of what? The light reactions of the photosynthesis. So it is like the 50-50 option now. So these two options we can remove. Non-cyclic, cyclic ತೆಗೆದುಬಿಡೋಣ, because these are a part of the light reactions. So we are left with only two options: substrate level, oxidative phosphorylation, right? Substrate level anta helidre enu, oxidative anta helidre enu. See, if the process of oxidation is taking place in the, see, oxidative phosphorylation can take place only during the process of respiration. Respirationally, in the presence of oxygen, if a particular molecule is getting oxidized, and because of the oxidation of that particular molecule, if the ATP is produced or GTP is produced, that is what is called as oxidative phosphorylation. But here we are talking about the Krebs cycle. Is there the presence or is there the participation of oxygen in Krebs cycle? Definitely no. That is why it will be substrate level phosphorylation. I hope you got the answer. 21. Signals of parturition originate from during the stage where the baby comes out from the mother's womb, mother's body, in the baby ಹೊರಗಡೆ ಬರ್ತಿರ್ಬೇಕಾದ್ರೆ. So the signals of parturition come from where? So the answer is both placenta and fully developed fetus. So a kind of reflex will be given, right, by the baby as well as the placenta, which is actually the signal for parturition. So the next question is, so the genetic problem, both male and female have a normal vision through their fathers, though their fathers were colorblind. Male and female ibru normal iddare, but our father color blind idru, and mothers did not have any gene for color blindness. So that means the parents of these male and females in the scenario, problem alliruvantaha male and female eniddare, so they are normal vision, they are having normal vision, and their fathers were color blind, mothers were normal once again. So they are asking, ee male and female cross ಮಾಡಿದಾಗ the F1 progeny ಏನು ಬರುತ್ತೆ, ಅದರಲ್ಲಿ how many daughters can become color blind? What is the probability of their daughters becoming color blind ಅಂತ ಕೇಳ್ತಿದ್ದಾರೆ ಅಲ್ವಾ? ಈ ಮೇಲ್ ಮತ್ತು ಫಿಮೇಲ್ ಅನ್ನ ಕ್ರಾಸ್ ಮಾಡಿದಾಗ ಡಾಟರ್ಸ್ ಬಂದವರಿಗೆ ಎಷ್ಟು % ಆಫ್ probability ಇರಬಹುದು color blindness, right? So see, you can actually the answer is 0%. ಯಾಕೆಂದ್ರೆ red and green color blindness in the X, it is actually X-linked trait, a recessive trait in human beings ಇರುತ್ತೆ. So a female with a recessive color blind gene will be actually only a carrier, but man will be affected, right? So here you can look at this problem. 0% answer. Nodi, parents of male and female. I hope this is visible for you. So here, the parents of male and female involved in the scenario, idu male, that is a father who is colorblind and mother is normal. When you take the gametes and carry out the cross, in the F1 progeny, andre male and female of the present scenario, illi carrier female irbodu, and male will be normal. Male will be normal and carrier female bantu, alva? So here, F1 progeny gives the genotype of male and female in the scenario. So, ಈ ಸಿನಾರಿಯೋನ ತಗೊಂಡು ನಾವು ಕ್ರಾಸ್ ಮಾಡಿದ್ರೆ ಬೋತ್ ಮೇಲ್ ಅಂಡ್ ಫೀಮೇಲ್ ವರ್ ನಾರ್ಮಲ್ ಅಂತ ಕನ್ಸಿಡರ್ ಮಾಡಿದ್ವಿ ಅಲ್ವಾ ಕ್ವಶನ್ ಅಲ್ಲಿ. ಸೊ ದೇ ಆರ್ ನಾರ್ಮಲ್, ಬಟ್ ಫೀಮೇಲ್ ಈಸ್ ವಾಟ್? ದ ಕ್ಯಾರಿಯರ್. ಸೊ ಕ್ಯಾರಿಯರ್ ಇದ್ದಾಗ ನೆಕ್ಸ್ಟ್ ಜನರೇಷನ್ ಏನಾಗುತ್ತೆ ಅಂತ ನೋಡಿದಾಗ ಕ್ರಾಸ್ ಮಾಡಿ ನೋಡಿದ್ರೆ, ಸೊ ವಿಲ್ ಗೆಟ್ F1 ಜನರೇಷನ್. ಹಿಯರ್ ಅಗೇನ್ ದ ಕ್ಯಾರಿಯರ್ ಫೀಮೇಲ್, ಸೊ ಕಲರ್ ಬ್ಲೈಂಡ್ ಮೇಲ್ ಅಂಡ್ ನಾರ್ಮಲ್ ಫೀಮೇಲ್, ನಾರ್ಮಲ್ ಮೇಲ್. ಅಂದ್ರೆ ಕ್ಯಾರಿಯರ್ ಫೀಮೇಲ್ ಇದೆ ಮತ್ತು ಕಲರ್ ಬ್ಲೈಂಡ್ ಮೇಲ್ ಇದೆ. The percentage or probability of color blind male is 25%, alva? Whereas carrier female ನೀವು ಏನು ಹೇಳ್ಬೋದು probability that is a 25%. But affected or color blind female ಅಂತ ಹೇಳುವಾಗ there is 0% probability. I hope we have answered this question. Is that clear? Now going to the 23rd one, the process of reverse transcription was brought to light by the work of. See, it is a direct question. You need to remember the name of the scientist here. So, yes, Archibald Gerard discovered alkaptonuria and studies its genetic. He studied the genetic basis of the disease. R.W. Hawley discovered the structure of RNA. He also shared the Nobel Prize with the two very well-known scientists, the Hargobind Khorana and Marshall Nirenberg, for describing the structure of alanine transfer RNA, tRNA of alanine, and linking DNA and protein synthesis. Whereas H.W. Temin and D. Baltimore in the year 1975, they discovered the process of reverse transcription. Reverse transcription was discovered by them, right? 24th one, answer is 4 here. 24th one, the diagram given here is representing the central dogma of molecular biology, and you have to identify A, B, C, D and E. So idralli nodi, I definitely you can answer this question if you know the simple concept of central dogma of molecular biology. And here you can say replication aguvantad yaudu, it must be DNA, right? And DNA, see here once again they are giving you replication here. So B, that means there should be RNA. So during the DNA getting converted into RNA, the transcription process takes place. D should be transcription, and here B, that is RNA, illi protein agbeku. C should be protein, and process is translation. The answer is what? C. Yes. 25th one, choose the wrong statements regarding the bacterial cell. So this is regarding the structure of the bacterial cell. Glycocalyx is the outermost envelope in bacteria. Yes or no? Yes, it is correct. The glycocalyx could be a loose sheath called capsule. How the? Let us see. The glycocalyx may be a thick and tough layer called slime layer. Actually, idu ulta agbekagittu alva. The glycocalyx tough iddaga it is called as capsule anta kariteve. It is loose layer idre it is called slime layer anta kariteve. Alva? So these two statements can be wrong. Let us go to the other statements. D, a special structure formed by the plasma membrane is called as mesosome. You know that the involved folding of the plasma membrane in the case of bacteria is called as mesosomes, and other similar structures like that of the mesosome are present. They are called as chromatophores. Chromatophores are known for holding the pigments in bacterial cells. Small bristle-like fibers sprouting out of the cell are called as fimbriae. So answer yavudu idralli anta helidre B and C are wrong statements. Rest other statements are correct. And you can also consider such statements are as informative. You can remember the information regarding that. 26th one, chloroplasts in the presence of high intensity of light align themselves in the mesophyll cell in such a way that their flat surfaces are. ನೋಡಿ, ಒಂದು ಸೆಲ್ ಒಳಗಡೆ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಇದೆ, ಪ್ಲಾಂಟ್ ಸೆಲ್ ಒಳಗಡೆ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಇದೆ ಅಂತ ಹೇಳಿದ್ರೆ, ದ ಮೂವ್ಮೆಂಟ್ ಆಫ್ ದ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ವಿದಿನ್ ದ ಸೆಲ್ಸ್ ವಿಲ್ ಬಿ ಸಚ್ ದಟ್ ದೇ ಅಬ್ಸಾರ್ಬ್ ದ ಮ್ಯಾಕ್ಸಿಮಮ್ ಅಮೌಂಟ್ ಆಫ್ ಲೈಟ್. not only they are absorbing the maximum amount of light, but they will also align themselves, they will also move inside the cell in such a way that they get protected. They are going to protect the chlorophyll present in them by getting denatured. Tumba highlight intensity ittu anta helidre adrinda protect ಮಾಡ್ಕೊಳ್ಳಿಕ್ಕೆ they will align in such a way that, so tumba highlight intensity iddaga they will be oblique, they will be not oblique, they will be perpendicular to the cell wall. If this is the cell wall, if this is the chloroplast anta helidre, so tumba highlight intensity ide illinda ee kade barta ide anta helidre, it will be perpendicular to this. Low light intensity alli it will be parallel to this, so that a larger surface of the chloroplast can be exposed to the light and it can absorb maximum amount of the sunlight. That's it. Okay, so answer is perpendicular to the cell wall. Tumba highlight intensity ide. 27th one, if Henle's loop were absent in mammalian nephron, which of the following is to be expected? ಏನಾಗ್ತಿರ್ಬೋದು Henle's loop absent ಇದ್ದಿದ್ರೆ? What is the function of Henle's loop? You have to understand. Henle's loop, the primary function of that is to reabsorb the water and sodium chloride from the urine. ಅಂದ್ರೆ ಯೂರಿನ್ ಇಂದ ವಾಟರ್ ಮತ್ತು ಸೋಡಿಯಂ ಕ್ಲೋರೈಡ್ ಅನ್ನ ಅಬ್ಸಾರ್ಬ್ ಮಾಡ್ಕೊಳ್ಳೋ ಮುಖಾಂತರ ಇಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಮೇಕ್ ದ ಯೂರಿನ್ ಅ ಲಿಟಲ್ ಮೋರ್ ಕಾನ್ಸಂಟ್ರೇಟೆಡ್. ಇಫ್ Henle's loop, if it was absent, then obviously what could have expected here? The concentration of the urine must have been little lesser than andre the urine will be more dilute. Answer is 1. 28th one, why asexual reproduction is sometimes disadvantageous? ಯಾಕೆ disadvantageous ಇರುತ್ತೆ? See, you have learnt about the inbreeding depression, right? So if any organism is allowed to if a plant is allowed to self-pollinate, or if an organism is allowed to undergo self-pollination or in the same species without any exchange of genetic material, then what happens? In the long run, it will undergo the deficiency or a kind of instability, or you can say biological impotence. That is what is called as inbreeding depression, and it produces genetically uniform population. See, this can be sometimes it can be beneficial, especially for high yielding varieties, but genetic uniformity in the long run may also cause reduced biological fitness in the organism, and that is what is called as this one. 4th is the answer. Contraceptive oral pills help in birth control by prevention of ovulation. So you have learnt about different concepts of contraception. That is the one. Down syndrome is an example for, you know that Down syndrome is caused because of what? 21st trisomy. You can look at this karyotype diagram. 21st chromosome, pair of chromosome nalli, there is an extra chromosome here, right? So that is why it is 21st chromosome. What is it? Trisomy, 21st trisomy, and the answer is aneuploidy of autosome. Aneuploidy means abnormal number of chromosome present in the autosome. That is a not sexual chromosome, not sex chromosome alla, autosome nalli present ide, because 21st pair of chromosome is a autosome. 31st one, cornea transplant in humans is almost never rejected. This is because, why? So it has no blood supply. Answer is what? It has no blood supply. Ali blood transfusion ಆಗ್ತಾ ಇದ್ರೆ, 2 different bloods are to be infused ಅಂತ ಹೇಳಿದಾಗ, then there will be blood clotting can take place, antigen antibody reaction ಆಗ್ತಾ ಇರುತ್ತೆ. When there is no blood supply at all, the problem is not there, so transplantation can be easier. So 32nd one, Jaya and Ratna were the high yielding varieties of whether rice or wheat or mustard or cotton. Direct answer, this is rice. 33rd one, a triplet codon means what? The presence of only 3 bases in mRNA, a sequence of 3 nitrogen bases in a tRNA, a sequence of 3 nitrogen bases in rRNA, a sequence of 3 nitrogen bases in mRNA. Which is the right answer? Yes, that is the 4th one, a sequence of 3 nitrogen bases in mRNA. Right? The next one, Alexander von Humboldt described the first time what? What did he describe for the first time? So it was species area relationship here. You know that this slope, species richness goes on increasing along with the area until a certain limit. And you should also remember this particular equation, log S is equal to log C plus Z log A. You should know the meaning of these terms in that equation. Yes. 35th one, respiratory quotient RQ is defined as what? RQ means what? So you have understood RQ for different biomolecules. For carbohydrates it will be 1 anta gottide, proteins it will be 0.9. So RQ for lipids will be always less than 1. Approximately you can consider 0.7 anta helbodu. RQ is nothing but volume of CO2 evolved and divided by volume of O2 consumed. So answer is this one, the ratio between CO2 liberated and O2 taken. So we can consider the next set of questions in the next session. Thank you.